Okay, good morning year 12. So this is just an introductory lesson into nucleic acids, and we're going to look at DNA and RNA. So we could put today's date, so it's going to be Llun 18 Ionawr, and then title DNA and RNA. And then I got two quick questions for you, stuff that you did last year at GCSE. Can we name the four bases? And why is DNA called a double helix? So get a quick pause and then we will go through the answers. Okay, so let's go through the answers. I'm going to pop them down here. Number one, can we name the four bases? So the four bases we looked at adenine, thymine, guanine, and cytosine. Okay, and then question two, why is DNA called a double helix? It's called a double helix because it's made up of two strands which are coiled or twisted into a helix shape. Okay. So what we're going to do then, we're going to state that deoxyribonucleic acid, DNA, is a polynucleotide, usually double-stranded and made up of nucleotides adenine, thymine, cytosine and guanine. Then we're going to look at ribonucleic acid, so RNA, and also that it's a polynucleotide, but it's usually single-stranded and made up of nucleotides adenine, uracil, cytosine and guanine. So let's have a look at our two types. So they're both nucleic acids, RNA and DNA. And as nucleic acids, they have the ability to carry instructions. We know that because DNA is made up of genes. And our genes code for our physical appearance, don't they? Or our physical characteristics that we have. So we know that they code for a set of instructions. Yeah, can't fit it in. Slide it over. Physical characteristics. Okay, and they have the ability to be copied, which we're going to look at in the next couple of lessons. Then we've got DNA and RNA are polymers. The individual nucleotides are the monomers that build up the polynucleotides. In DNA, it's deoxyribonucleic acid, and in RNA, it's ribonucleic acid. When we look then at what a nucleotide is, so this is going to be the monomer. Nucleotides are made up of three smaller components. We've looked at this already, so it's nothing new. We have a phosphate group, okay, which contains, we've got our phosphate group. Then it is attached to our sugar, depending on if it's RNA or DNA, you might get a ribose sugar. And then we have our base attached. From there then, if we look, we've got a polynucleotide. So we've got the sugar and phosphate units make up the backbone of the nucleic acid. So we know, I'm just going to highlight now. I'm going to highlight there. Yep. I'm going to highlight in red, okay, that this is classed as the backbone. And it's the backbone because it makes the strand, okay. Then we have here, I'm just going to highlight it because it hasn't come out so well as I've done it. We've got one, we've got all of these, and these are our bases. And our bases will bind to each other to get our second strand, if we're looking at, say, DNA, for example. And then if I highlight, my pink highlights, okay, one nucleotide would be one phosphate, one sugar and one base together. That would then make one nucleotide. But we know this is a polynucleotide because, as you can see, I have many nucleotides joined together. And now we're going to look at how they are joined. Okay, so the two strands on top of helix are held together by hydrogen bonds. So as you can see here, we have, let me get my thing. We have cytosine, binding guanine, adenine and thymine, and we call that our complementary base pairing. And we know, and we have known, that adenine always binds with thymine and cytosine always binds with guanine. But we haven't looked at the bond, and the bond that holds them together, or bonds the strands together, is hydrogen bonds. And we know about hydrogen bonds from our first topic. Okay, hydrogen bonds are quite weak, but collectively they are very, very strong together. Between cytosine and guanine, we have 3 hydrogen bonds, and then between adenine and thymine, we have 2 hydrogen bonds. Connecting our phosphate and our sugar together, then we have a condensation reaction. And if we're not sure, if we can't remember what a condensation reaction is, okay, essentially it's a reaction where we remove H2O for them to join together. Okay, so our bases then, there are 5 different nitrogen bases. We've got adenine, thymine, uracil, guanine, and I'm missing cytosine. Don't know what's happened to my PowerPoint, but it's okay, we can fix it. Okay, when we look at translation and transcription, you will notice that when we look at DNA replication of them, that we have adenine and thymine, guanine and cytosine in DNA, but when RNA replicates our DNA, thymine is replaced by uracil. So in RNA, our complementary base pairing would be adenine will bind with uracil, and then cytosine and guanine still together. Okay, it's the only difference that's in RNA because there is no thymine. Okay. These bases then, we have different types of bases. We have larger ones and we have smaller ones. So if we are looking, adenine and guanine are our larger bases. They're called purines. Okay, and these have double rings of carbon and nitrogen atoms. So if you can see from the diagram, just move it out, that we have the double ring. Okay, so essentially they're a lot bigger. Okay, and then we have our smaller ones, which are thymine, uracil, and cytosine, and they are pyrimidines. So they have a single ring of carbon and nitrogen atoms, and they are smaller. That's why they complement each other quite well in the base pairing. We have one large and one small pairing together. If we then look, okay, we know that they are made up of strands of alternating sugars and phosphates, but the strands run in opposite directions. So one's going upwards, one's essentially going downwards, and they are held together by hydrogen bonds between the 2 strands. So if we were to look at it, okay, we have one strand. If I highlight my one strand, so we start with our 5, okay, and it goes my one strand, and it ends with my 3. Let me just zoom out slightly because that hasn't worked so well. Okay, and then we have the other strand, okay, and it's going the opposite direction, where we have 5 on this side, and our 3 on the other. Okay, and that's what the 2 ends of DNA are called. They're called our 5 and they're called our 3 ends. Okay, but they go opposite directions for us. So what I would like to do now, if you want to pause the video, and can you name me the 4 bases of DNA, the 3 components of a nucleotide, the 2 ends of a strand are called, and one place where a condensation reaction occurs. And then for a bonus, can you tell me the difference between purine and pyrimidine bases? Okay, so let's go through the answers. I'm going to pop them down here. So we should have had for our 4 bases, nice and easy, it was in our starter, adenine, thymine, guanine, and the cytosine. Okay, number 2 then. Okay, if I number these because it's gone the other way, I'm probably getting confused. 2, the components of a nucleotide. So nucleotide, then we have a phosphate group. We have a sugar, and it's a pentose sugar. So there's 5 carbons. And then we have one of our bases. Number 3, the 2 ends of a DNA strand are called, they're called our 5 and our 3 ends. And then number 4, the place where condensation reaction occurs. Condensation reactions occur between a phosphate and a sugar. Okay, in the backbone of the DNA strand. If we then look, okay, so the bonus question, we've got describe the differences between purine and pyridine bases. So I'm just going to do it as a quick table. Purine and pyridine. Okay, so purine are larger in size, pyridine are smaller. Okay, purine have 2 rings of carbon and nitrogen atoms, whereas pyrimidine has 1 ring of carbon and nitrogen atoms. And then we can name the bases. So we have adenine and we have guanine, and then we have thymine, uracil, and cytosine. Okay, so let's have a look then. We said that the strands were in opposite directions. So for the DNA strand, TACG, ATC, AT, the correct complementary DNA strand is, so have a little look now. I will just highlight DNA for you. So we're going to be looking at a DNA strand. And can you work out which is the correct one? I'll give you a minute to have a think, and then I'll go through the answer. Okay, and the correct answer would be B. Okay, so it's B because we're given the opposite ends. We start with a 5, which is going to be the 3, and we end with a 5, which they all do, lovely, and then we can sort of swap them over. So T to A to T, etc., etc. Okay, when you're looking, okay, we could have thought C, and it would have been C if the question asked us if we're looking at an RNA strand. Okay, so C would have been correct if we're looking at a complementary RNA strand, not a DNA strand. Okay. So we've got, what is the function of DNA? So hereditary material responsible for passing genetic information from cell to cell. We know that it's got all of our genes on. It's got every bit of information about us on there. So how is it adapted to do its job? Very stable. Okay, the 2 strands can separate, so it can self-replicate, which we're going to look at next lesson. The large molecules that carry lots of information. The base pairing prevents corruption from outside chemicals or physical forces, because adenine won't bind with anything else. Okay, same with cytosine, same with guanine and thymine. So our 2 scientists, which we're going to spend a lot of time looking at over the next couple of lessons, Watson and Crick hypothesis. Okay, we've talked about them before in year 11. So Royston and Franklin in the early 50s showed that the phosphate groups of DNA must lie on the outside of the molecule. Watson and Crick put forward a possible structure of DNA in 1953, and they were awarded a Nobel Prize in 1962. So, questions for you to answer, which I will then go through. So what elements is DNA made of? What are the monomer units called in DNA? Name the 3 parts of the monomer. Which bases form complementary base pairs? What holds the 2 strands of DNA together? If the base sequence of one strand of DNA is GTT, ACC, GTA, what would a sequence be on the other strand? If there's 19.9% of base pairs in DNA are guanine, what percentage is thymine, and then why is DNA suited to do its job? So press pause, have a go at the answers, and then I will go through it. Okay, so let's go through the answers. What elements are DNA made of? So mainly made up of carbon and nitrogen with phosphates. We've got the phosphate group. Okay, question 2. What are the monomer units called in DNA? They are called nucleotides. Then name the 3 parts of this monomer. We've gone through this already. Phosphate, a sugar, and a base. Question 4. Which bases form complementary base pairs? Nice and easy. Adenine to thymine, cytosine to guanine. Question 5. What holds the 2 strands of DNA together? Well, it's the bases, and then we have hydrogen bonds. Ooh. Hydrogen bonds, excellent. And then question 6, if the base sequence of one strand of DNA is GTTA, what will the sequence be on the other strand? So it'd be C, A, T, G, G, C, A, T. If 19.9% of the DNA's base pairs in DNA are guanine, what percentage is thymine? So this is where we can get caught out, because you'll think, well, it's just the opposite. So if you've got 19.9% equals guanine, we are then going to have 19.9% that is cytosine. And the reason we're going to have that is cytosine because they're complementary base pairing. So if 19.9% is guanine, we have to have the same amount of cytosine because they pair together. So in total, that would be, this is where my maths is going to be dodgy, 39.8%. So out of a possible 100%, we're going to minus that. Right, oh God. So 216. So we have 61.2% left. Now we're going to look at that thymine, but out of that has to be divided by 2 because if we've got so much of thymine, we have to have the exact same amount for adenine. So if we're going to divide that by 2, we would have 30.6% of thymine and then 30.6% of adenine. We would have the exact same. So our answer, 30.6% thymine. Really good question. It's a question where we can get confused and we can lose really, really easy marks. Okay, because straight away in the exam you panic and you think just the opposite. You would have probably give me an answer of 80.1%. Okay, because you just like taking the 19.9 away from 100%. And then why is DNA suited to do its job? It's suited to do its job because it's stable. Okay, it's also can self-replicate. It's a large molecule, so it can hold lots of information. And then we have that the base pairing prevents corruption from outside events. Right, Lovely, so that was just a quick lesson. Next lesson then we'll look at translation and transcription, so how does DNA replicate is what we're going to look at. Right, any problems, let me know. Thank you, Year 12.